Good morning, students. This is Sheba. Now we are going to discuss about the EC8073 medical electronics. Before getting into the topic of discussion, first let us get a brief intro of what is meant by this medical electronics, why we need to know about this. So regarding this paper, it combines medicine as well as the engineering field, how our engineering knowledge and technology is applied in curing and diagnosing many diseases in medical field. So here it is used to gain knowledge about the various physiological parameters, both electrical and non-electrical, using method of recording and also method of transmitting the parameters. And also we learn about various assisted devices that are used in hospitals. Regarding the syllabus of medical electronics, here you will be about 5 units, where Unit 1 covers electrophysiology and biopotential recording, and Unit 2 covers the biochemical and non-electrical parameter measurement. Unit 3 discusses with assisted devices, and Unit 4 we deal with physical medicine and biotelemetry. Finally, Unit 5 combines recent trends in medical instrumentation. So here I have also given the suggested textbook and reference books for clarification purpose, and also NPTEL lecture material references are also given, which combines both video as well as the PDF materials. Moving to the introduction part, what is meant by this medical electronics? It is a scientific direction in technical electronics which aims to develop electronic devices for experimental, preventive and clinical research and treatment of human. If you see the application sector of this medical electronics, it is applied in many real-time applications like digital thermometers, digital stethoscope, BP measurement, sugar level monitoring equipment, MRI CT scan equipments, heartbeat monitoring equipments, X-ray scanning device, etc. So everything in medical field combines this electronic equipments. And so the role of this electronics in medical field is an inevitable one. Regarding the medical instrumentation, biomedical instrumentation is nothing but it is a field of creating such instruments that helps us to measure, record and transmit data to or from the body. So it measures many parameters like temperature, displacement, biochemical parameters, etc. There are many types of medical instruments available like direct or indirect device, invasive, non-invasive types, contact, remote sensing device, sense and actuating device, real-time and static device. Moving to the original of biopotentials. What is meant by this biopotential? Why we need to measure the biopotential? Generally, if we need to measure any parameter, first we need to find the signal emitted by the device. For example, if you are measuring a PN junction diode, means you will be finding the output voltage. So how much voltage it gets as the input and how much current it produces as the output. So we will be getting the VI characteristics to find the output. Similarly, the human body also emits some potential, and we need to measure the potential in order to find the state of the human body, whether it is in normal condition or in abnormal state. And so this medical electronic deals with a recording of potentials like electrocardiogram, ECG, electroencephalogram, EEG, electroneurogram, ENG, electromyogram, EMG and electroretinogram, ERG. Talking about these biopotentials, they are known as excitable cells, in which it acts in two conditions. One is resting state, another one is action state. So biopotentials are nothing but an electrical potential that is measured between points in living cells, tissues or organisms, and which accompanies all biochemical processes. So you can clearly see the diagrammatic representation of this biopotentials, how it is measured from the human cell or tissue, and it is converted to the potential value, and it is drawn as a sinusoidal graph, which you can clearly see Vm across time duration. So based on the mechanism, these biopotentials are calculated based on the ion concentration in our tissues. So taking the concentration of ions within the human body tissues. So if you are selecting a cell portion, the inside of the cell will be mostly surrounded by potassium ions and outside will be sodium ions. So based on the concentration levels, whether it is in a neutral condition or any unbiased values occurs, based on the ion concentration values, we find the value of potential. So Nernst and Goldman equation are corresponding equations used for finding this biopotential values. Talking about these biopotentials or cell membrane potentials, we need to find the ion concentration, whether it is in the equilibrium neutral state or in abnormal condition. So if you are giving any excitation to the human body or to any human cell, the ion concentration will suddenly start varying and it creates the action potential. Whereas when you are not giving any input, means it will be in the idle resting condition. So similarly, in the resting potential can also be calculated, which is nothing but equilibrium potential based on the Nernst and Goldman equation. Under this resting condition, the excitable cells maintain a steady state electrical potential difference between the internal and external environments. And the membrane here is slightly permeable to sodium ions and freely permeable to potassium and chlorine ions. So, talking about this active and resting potential, a human body can be changed from resting to action potential by giving any external stimulation or excitation. So, for example, if you are recording electrical potential value from a nerve fiber, cell membrane potential versus time duration is taken. First, you will be finding the resting potential, and also you will be noting down the action potential after giving this certain excitation. Action potential also includes two major parameters like absolute refractory period, relative refractory period. What is meant by this refractory period? It is nothing but the membrane cannot respond to any external stimulus and is said to be the absolute refractory period, whereas the action potential can be elicited by an intense superthreshold stimulus, which is said to be the relative refractory period. So based on this concentration of ions and volume conductor experiment, we can find the value of potential variation between the human body. And these results record the surface potential whether the nerves are excited or in the idle condition. There are also organization of peripheral nervous system, which includes the reflex arcs and sense organs, from where you can find the different potential values. We can record different values of excitation of the human body, which is clearly depicted in the diagram. And also junctional transmission, that is, you will be choosing a portion of junction where you will be finding the value of the ion concentration and how the junction reacts. For example, if you are finding the concentration of blood in a particular portion, for example, take hand. If you are going to find the concentration of blood in the hand portion, then you will be reflecting and light through the blood. If the light is observed and only minimum amount of light is reflected, then you will be check whether there is any problem in that area. If entire light is reflected without any absorption, you can compare it to the reference value whether it matches or not. If it matches, then there is no problem. Like that, you can also use this experiment for finding the value of blood content. So talking about this introduction part, hope you have got a clear knowledge about what is meant by this biopotentials, how we are measuring them. So you need to clearly know about the resting potential, action potential, conduction velocity, what is Nernst equation, Goldman equation, how they are used to find the value of potentials in the human body. Talking about the part A, part B, part C questions, you need to know the clear outline of what is meant by this biopotential, how in real time it is used to measure the parameters of human body. Here I have also given the important links for reference, website link as well as the book reference, corresponding book SP Sasireka Medical Electronics, page number, chapter 1 to 6, page number corresponds to the topic which we have discussed now, and the content of presentation is given for your reference. So talking about the real-time application of medical electronics, as I have said in the earlier part, this medical electronics is applied in many areas. For example, you can take a simple example of ECG. Everyone will have seen this either in the television or in the real time. ECG measuring. A patient will be admitted in the hospital. Their heartbeat is monitored, based on the heartbeat variations, we will be finding whether the patient is in normal condition or in the abnormal condition. So it is a real-time example of our medical electronics. So our engineering knowledge is applied in the medical field, and in everyday life it is developing to its extreme based on the types of diseases evolving. You can see day by day there is a lot of changes in the diseases coming, and so medical equipments need to be upgraded to a level which can cure a lot of disease and save the mankind. This is about the topic which we have discussed now. If you have any doubt in this content, you can ask me and get the details. Thank you.